So I should start sharing, or yeah.
I'll just introduce you. Okay, so good afternoon, good morning, good evening to people wherever they are. Today we have Dr. Terence Crofts from Northwestern University. Dr. Crofts is a research assistant professor at Northwestern. And before this, he did his postdoctoral fellowship at Washington University in St. Louis. And before that, he did PhD at University of California at Berkeley. He's an accomplished researcher with a lot of students trained under his mentorship. And he basically works on antimicrobial and bacterial interactions and how those two things evolve and basically respond to each other. So we'll hear more about it from Terence, and take it away.
All right, thank you very much for that introduction. I'll start sharing the slides. So I think it should be showing up well. So again, thank you for the introduction. I'm a research assistant professor at Northwestern University, and I'm going to be talking today about my research studying outside the box antimicrobial bacteria interactions, as I like to think about it. And so just to give a preview for the talk, I'm going to begin by talking a bit more about what I mean by these sort of outside the box interactions and why we should care about them, especially in the case of antimicrobials. And I'm going to go over three recent stories I've been working on illustrating these, with one project concerning how housekeeping enzymes can evolve into resistance enzymes potentially. Another story dealing with bacteria that don't only resist antibiotics, but actually break them down into carbon sources. And then a third story looking at this really neat method and some modifications that I've made to that method that kind of open up the studying of microbiomes as a whole for these sort of interesting antimicrobial microbiome interactions now. And so to begin with a bit of background, I think a fun way to do this, so to speak, is to jump back about 350 years into 1665 London. And this is actually the year that Isaac Newton spent at working from home and developing calculus, as lots of folks try to remind us for this year. Another work from home year. What I'm showing you here is this really neat sort of census, if you will, called the Bills of Mortality. And basically what this shows is that for every week, people tallied up what the causes of death in 1665 London were. As I mentioned, this was a plague year, so there were a lot of deaths due to that particular microbe. But if you look in general at some of the causes of death in London, you can see that a lot of these could be attributed not just to infectious disease, but in many cases, probably specifically to bacteria. So you have things like the plague, of course, but you also have flux, like the dysentery, gangrene, Clostridia species, consumption, which might be tuberculosis. And so, you know, we have a pandemic year, but at least we don't have all these other bacterial illnesses to worry about. And that's thanks largely in part to these miracle compounds, antibiotics. What I'm showing you here is this timeline. What I want you to focus on is this so-called golden age of antibiotic discovery, centered in the 1930s to 1950s. And it was during this period that most of the natural products and scaffolds that make up our modern antibiotic armamentarium were discovered. What's really interesting is you can see that in blue on this timeline is the period of time where these antimicrobials were essentially uncontested against infectious disease, whereas red indicates the time period where bacteria began to develop antibiotic resistance. You can see that while a lot of these compounds are still being used today and are still very effective, in most cases, there was a very quick development of antibiotic resistance to these antibiotics, which is obviously problematic because most of modern medicine is built around these compounds, not just for curing infections, but for also making compromising procedures possible. Things like chemotherapeutics to treat cancer or organ transplants, these all depend on having sort of a backstop of antibiotics to treat potential bacterial infections. And so this graph really kind of illustrates how this is a problem, because while in recent years, fewer and fewer large pharmaceutical companies have been researching new antibiotic treatments, there's been at the same time, this increase in the incidence or rate of infections that are due to antibiotic-resistant bacteria. So it's estimated by the year 2050, we'll see something like 10 million global annual deaths due to antibiotic-resistant bacteria. So that's really surprising and scary because it means that just a few decades ago, those deaths would have been entirely avoidable because those bacteria would not have developed antibiotic resistance yet. And so why I want to talk about this in sort of a outside the box perspective is because we know a decent amount about the antibiotic resistance genes that are going around clinics now. But what we're doing across the world is this kind of uncontrolled global Darwinian evolution experiment where we're essentially assaulting microbiomes within our own bodies and the environment with antimicrobials and other pharmaceuticals that are putting selective pressure on those communities and bacteria to develop enzymes that will resist or modify those compounds. And to kind of visualize that, this is a nice photograph of a wastewater efflux pipe from a pharmaceutical plant that drains into a local body of water in Southeast Asia, which kind of nicely illustrates that this idea we're flinging these antimicrobials and other pharmaceuticals into the environment and developing what's going to become the next set of antibiotic resistant mechanisms that will reach the clinic eventually. And so that's what I mean by how I want to look outside the box. These are things that we don't really know yet. They're just sort of emerging. And again, the reason why this is likely to happen is that when we're throwing these antimicrobials and pharmaceuticals into the environments, they're hitting these ecosystems called microbiomes, which are assemblages of exceedingly diverse microbial life, including a lot of bacteria. And so these contain a very high diversity of genes, biochemical functions. And so it's basically a perfect environment for the development of new antimicrobial resistance genes. And these microbiomes, of course, exist in that body of water that the pharmaceutical plant was draining into, but also includes things like our household surfaces, our own gut microbiomes, and livestock. And so this is where those worry new resistance genes are going to come from. And so what I want to talk about is my general interest, which is discovering these new resistance mechanisms or new pharmaceutical modified enzymes from the environments before they have jumped into the clinic. And so this kind of takes the form in today's talk of studying where antimicrobial resistance might come from, studying other forms of bacterial antibiotic or antimicrobial interactions, in this case, catabolism, and then also developing new methods for studying all these different interactions. And so with that background, I'm going to jump into the first project that I want to talk about today, which concerns these enzymes that have been dubbed chloramphenicol reductases, which are perhaps a nice illustration of how housekeeping enzymes can evolve into antibiotic resistance enzymes. And so this kind of goes back to that figure I showed earlier, where we have seen this rise of antibiotic resistant bacteria and flourishing of antimicrobial resistance mechanisms. And so there's this question of where these come from. And for the most part, when we talk about these, we're talking about clinically sort of professional antibiotic resistance enzymes. And these are usually characterized with this macrolide antibiotic as an example by being enzymes that are close to kinetically perfected for their function. So they have very fast substrate turnover. They have high affinity for their antibiotic substrates. Oftentimes, the genes for these enzymes are induced by antibiotic, and these enzymes can be spread easily. We have a decent sense that many of these enzymes might actually originate from bacterial producers of antibiotics that use these enzymes as self-protection mechanisms. And so again, these enzymes have fast turnover, high substrate specificity, but really these are kind of the known knowns of the antimicrobial world. What I want to talk about are the kind of unknowns, where there are enzymes out there that can use antibiotics as substrates, but they haven't gone to the clinic yet, but it's still worth studying them and knowing if they have the potential to evolve into these professional antibiotic resistance enzymes. And there's this idea that a lot of housekeeping enzymes in bacteria, for example, have the potential to make this evolutionary jump to a resistance enzyme. Why that's interesting is because in contrast to these sort of professional enzymes, these housekeeping enzymes have much greater substrate promiscuity, so they don't bind antibiotics particularly well. They have slower substrate turnover. They're generally not induced by an antibiotic and are constitutively expressed by bacteria. And so what I want to talk about is this really interesting idea of how housekeeping enzymes might become resistance enzymes. And to do that, I'm going to focus on this antibiotic called chloramphenicol as a model antibiotic for this. So chloramphenicol is really neat. It's one of those antibiotics that was discovered back in the golden age of antibiotic discovery. And it was used very successfully clinically up through around the 1980s, when its use was discontinued because of increasing toxicity in the US, Europe, and several other countries. But nonetheless, it still remains one of the World Health Organization's essential medicines. And so when we think about chloramphenicol resistance, we usually think about the mechanism that is seen most often in the clinic right now, which is via chloramphenicol acetyltransferase, or CAT enzymes. And these essentially modify these hydroxyl groups on chloramphenicol by adding an acetyl group, and that prevents the antibiotic from binding its ribosome target, which therefore spares bacteria its effects. But I want to jump back to just a couple of years after the discovery of chloramphenicol to discuss the first mechanism of resistance that was discovered to this antibiotic, and that is enzymatic reduction of the drug, which is found to be nitro reduction of the nitro group. It has been carried out by diverse bacteria, including Strep pyogenes, or E. coli, and several other bacteria. And why this is interesting is that in a lot of these cases, it seemed like this modification was giving just a kind of low level of resistance to the antibiotic, which suggests that it could be a good model for a housekeeping enzyme that has antibiotic resistance as a side activity. And so I started this project by trying to look for what genes or enzymes might be responsible for this. There was a candidate that really jumped out, which is this enzyme family called NFSB, for nitrofurazone susceptibility locus B. What's interesting about these enzymes is that other folks have studied them and shown that it's a housekeeping enzyme that has a lot of substrate promiscuity. So I'm showing you here is just a figure illustrating that this enzyme can actually use this other pharmaceutical, a nitrodiazepine antipsychotic, as a substrate and reduce it to produce a kind of similar product to what people had seen with chloramphenicol earlier. And so again, this NSFB enzyme has this very high substrate promiscuity, like a housekeeping enzyme. People see that it has slow kinetics with these aromatic nitro substrates. It's not induced by antibiotics, and it is a gene that is found throughout microbial taxa on the chromosome. Really, again, hammering home this idea that it is a housekeeping gene. And what I'm showing you here is the evidence that it's not just a housekeeping gene, but it's a housekeeping gene that can give resistance to chloramphenicol. So what I did was I went through the literature and found candidate NFSB homologs from each of these different bacterial taxa, which have been shown in the literature to have the ability to reduce chloramphenicol. So I took the NFSB homologs from these and expressed them in E. coli, and then tested that E. coli's change in resistance to chloramphenicol. So this is a dose response curve where you can see how much the E. coli strain expressing these NFSB homologs grew in response to how much chloramphenicol was added to the culture. So the empty vector control, shown in empty black circle, is almost entirely inhibited by 4 micrograms per mil chloramphenicol. The control strain expressing an acetyltransferase is essentially immune to the drug, but then each of the strains that's expressing an NFSB homolog shows this very nice step up in resistance to chloramphenicol. And this can be summarized by taking the concentration of chloramphenicol required to inhibit the growth of these strains by 50%, and that's shown here. What really stood out to me of this data was that while NFSB expression from some of these strains showed a little bit of an ability to change the susceptibility of E. coli to chloramphenicol, E. coli expressing the H. influenza NFSB had a markedly increased level of resistance. So this is a really great example potentially of, again, an NFSB housekeeping enzyme giving resistance. And so one way to kind of establish if it's a housekeeping enzyme in this case and not actually a resistance gene is to look at the enzyme kinetics of this reaction. So I'm looking at the reduction of the nitric group of chloramphenicol to an amino group. And when I measured that enzymatically, I got this very nice, kind of classic Michaelis-Menten enzyme kinetics curve. We can see as I added more and more chloramphenicol as a substrate to these reactions, I saw an increase in the initial rate of the chloramphenicol reduction activity, which eventually plateaus out. So this is a very sort of classic, nice-looking enzyme substrate interaction. So this is a housekeeping enzyme, and it really is using chloramphenicol as a substrate. But if you look at the catalytic efficiency, or the Kcat over Km metric for this enzyme with chloramphenicol, it maxes out about 2 × 10⁴ per molar per second. I put that up there because, in contrast, most antibiotic resistance enzymes, these professional enzymes, instead max out at 10⁷ or 10⁸ per molar per second. So this is a real enzyme substrate interaction, but it really is a sort of moonlighting side activity for the enzyme, confirming that this is really a housekeeping enzyme that happens to have this neat ability to reduce chloramphenicol and maybe has the ability to evolve into a real resistance gene later on. And just to follow up on that, I use LCMS, mass spectrometry, to again confirm that the chloramphenicol substrate can be converted into an aminochloramphenicol product. This is the chromatogram of an enzymatic reaction, showing that these two peaks match up with the chloramphenicol and aminochloramphenicol standard. When I look at the actual mass spectra of this peak, can again confirm that the fragmentation pattern matches aminochloramphenicol. And that's important because if this enzyme activity did not actually reduce chloramphenicol all the way to an amino group, but instead reduced things like partway, bacteria would have a problem because it actually takes sort of 3 rounds of reduction to get from a nitro to an amino group. And if you don't do all 3 of those reduction rounds as a bacterium, you end up with unstable intermediates that can spontaneously back oxidize the chloramphenicol. And so you can have an enzyme that reduces chloramphenicol just 1 step, but it won't give resistance because they'll just allow that product to back oxidize into the antibiotic again. So it's significant in this case that we actually did see aminochloramphenicol as a final product, which kind of led to this interesting question of, are there any other antibiotics out there that have this sort of nitro group? And there's a really good example to be found in this drug called metronidazole, which again has this nitroaromatic group. And interestingly, when we look at just the vector control E. coli, it's largely resistant to metronidazole. And in contrast, when you start having it express NFSB homologs, including the H. influenza homolog, instead of giving the resistance, you're increasing the susceptibility of E. coli. And that's because metronidazole and other drugs like it are prodrugs. It's not actually toxic until a nitroreductase tries to act on this. That results in the prodrug being activated to its toxic form, which kind of leads to this kind of interesting idea of, could metronidazole and chloramphenicol essentially trap E. coli in the catch-22 if it's expressing a chloramphenicol reductase, NFSB. And this figure shows that you do indeed get this sort of catch-22 for the E. coli. This is a just a figure showing the results of a set of experiments where I grew the E. coli expressing the H. influenza NFSB in the presence of a combination of metronidazole and chloramphenicol in a so-called checkerboard assay, which at these points is a combination of these 2 drugs. And basically, we can tell by the fact that this curve is below this dashed line, and that this so-called fractional inhibitory concentration index is less than 1. These 2 things tell us that chloramphenicol and metronidazole are synergistic in this strain, which kind of lets me show this nice summary of things where this illustrates nicely what I mean by this being a catch-22, where if the E. coli is expressing this nitroreductase, that by itself is sufficient to give it resistance to chloramphenicol. So it takes this active antimicrobial, reduces the nitro group to give you an inactive product. But at the same time, that very same nitroreductase can take metronidazole as a substrate in its prodrug inactive form inactivate it to its cytotoxic product. And so this is a really cool example of how you could set up basically a competing system in bacteria that resist chloramphenicol by nitroduction. Suggests that they'd be more susceptible to drugs like metronidazole. So I want to show next is a new project that kind of builds on that first one, trying to look at how widespread is this chloramphenicol reductase activity. And what I did was I went out and found a series of more closely related NSFB homologs, as well as more distantly related ones. And what I'm showing you here is a phylogenetic tree for those enzymes, where we have a series of H. influenza NFSBs from different strains. And so these basically range from 99% identical to the NFSB I've been talking about from the KW20 strain, down to 95% identical for this strain 12 NFSB. And then more distant related are these Neisseria NFSB and this Pastorella NFSB. And these are about 60% identical to the KW20. And so what it nicely does is it gives this range of relatedness that can try to match up to activity in order to see, first off, what are these sort of sequence function relationships of these enzymes, and also try to see what the functional landscape of NFSB enzymes is like, and is there any evidence that it could be having the potential to become a real resistance enzyme. And so I show you here again is an experiment where I expressed all of these NFSB homologs in E. coli, and in every case, except for Pastorella, expression of these homologs once again gave E. coli resistance to chloramphenicol to varying degrees, showing that this enzyme class very broadly across Haemophilus and Neisseria taxa at least has this potential to evolve into a resistance mechanism. And so I'm showing now is some data that I got in collaboration with some folks at Argonne National Labs. And so we purified the enzymes from most of these new NFSB homologs, all four of the Haemophilus influenza strains, as well as the Neisseria and meningitis NFSB. And once again, we found that in every case, these enzymes are able to convert chloramphenicol to immunochloramphenicol. And interestingly, when we look at their kinetics, they have a very different kind of maximum rates and Km, sort of a binding measure for chloramphenicol. So I'm showing you here again is that Michaelis kinetics showing that all these enzymes are using chloramphenicol as substrates. Really interestingly, this NFSB from H. influenza strain 12 is significantly superior to all the other NFSBs at reducing chloramphenicol. And so it's really interesting again because compared to the other Haemophilus influenza homologs, this enzyme shares between 95% and just over 96% homology, which really sets up this nice situation where we can look at how the sequence of this enzyme differs from its close homologs and how that might explain this substantial jump in chloramphenicol reduction activity. And so what I'm showing you here is a multiple sequence alignment. And it turns out that the NFSB from strain 12 differs from the rest of the Haemophilus influenza homologs at only about a half dozen sites. We solved the crystal structure with my collaborators at Argonne National Labs, and most of these residues are not near the binding sites. We don't think they're important. And this methionine to isoleucine change is also probably not very significant, but what's really suggestive of being important is this serine to proline change. So again, the HI12 NFSB has a serine here compared to a proline in its related homologs. Those, of course, are very different amino acid residues. And what's really neat is that we can look at the active site of the structure that we solved and see how this might make sense. And so what I'm going to point out here is this tryptophan residue at position 71. It's actually known from other studies that having a bulky aromatic amino acid at this position is important because it helps act sort of as a lid to keep substrate in the binding pockets near to the FMN coenzyme, which is where a lot of the chemistry happens. And what's significant is that this position 68, which is a serine in our very active HI12 homolog, but a proline in the less active homologs, is positioned right here to essentially, we think, act as a hinge for opening up that binding pocket capped tryptophan. And what we think is happening is that in the less active enzymes, this is a proline that has a lot less flexibility, which probably slows down the turnover that can occur in this active site. But by having that flexible serine, the H. influenza 12 strain has kind of a much more rapid turnover. And we think that might be why this enzyme shows so much more activity with chloramphenicol. And so we're really interested in looking at a wider array of crystal structures in the future and trying to figure out what residues might need to change in these enzymes for them to go from being housekeeping enzymes that can confer resistance towards being a real antibiotic resistance enzyme that could evolve. And so to kind of sum up this first story, I'll first mention that again, we found that there are these NFSB enzymes or genes that when expressed in E. coli can give resistance to chloramphenicol. And really interestingly, these seem like a really good candidate for studying housekeeping enzymes that have antibiotic resistance. And we showed that because the kinetics of its chloramphenicol reduction show a real substrate enzyme interaction, but it's not at the level of a professional antibiotic resistance enzyme. But we are working on identifying the key residues that might allow that housekeeping enzyme to jump into becoming a resistance enzyme. And so with that, I want to switch over a little bit from talking about just antimicrobial resistance to instead kind of talking about the ecology of antibiotics and how we can kind of look at that relationship with bacteria in a new sort of way, specifically where antibiotics are a carbon source potentially. And so this kind of gets back to this idea of antibiotics being a natural part of soil ecology. What I mean by that is that, I mentioned that antibiotic resistance has been increasing in recent years, but folks have actually very elegantly looked essentially back in time and shown that antibiotic resistance is ancient. These experiments have taken the form of other groups digging under permafrost down to sediments that were laid down 30,000 years ago and finding antibiotic resistance genes, or other groups that have looked at cave systems that have been sealed off from the outside world for millions of years, and again, finding antibiotic resistance enzymes in those caves. So this really shows that resistance existed before humans started overusing antibiotics. So antibiotics are a natural part of ecosystems. And this makes sense because we know that a lot of antibiotics, and in fact, a lot of pharmaceuticals that we use, are actually so-called natural products. They're derived from bacteria or fungi that are found in things like the soil microbiome. So we know that a lot of these antimicrobials are, again, natural compounds in these ecosystems. And so we know that they're made there, and we know that they're resisted in the soil, which kind of opens up this question of how do you close the carbon cycle of these compounds? How are they recycled? And so that gets back to some really interesting research that was started again in that golden era of antibiotic discovery, where scientists started to find out that bacteria can actually break down and use these compounds as carbon sources. And so I want to take this quotation from this 1951 paper about streptomycin, where the authors wrote, "Nevertheless, the fact that streptomycin is a natural product gives reason to believe that it can be destroyed by microorganisms." And so it's very nicely summed up this idea that antimicrobials and antibiotics must be consumed and recycled in nature somehow. And so more recently, my postdoc advisor during his postdoc went out and performed a more high throughput search for antibiotic metabolizing bacteria. And what he and his collaborators found was that for every soil, like soil F1, F2, F3, that they surveyed, they were able to isolate bacteria that could use at least one antibiotic as a sole carbon source. And for every antibiotic that they looked at, they could always find at least one, oftentimes several different bacterial isolates that could use that antibiotic as a sole carbon source. And so when I started this project, we still didn't actually know how any of this really happened. We just know that it did. And so I decided to focus on this antibiotic, Penicillin G, in part because this is one of the antibiotics that seem to be most commonly metabolizable by soil isolate bacteria. And so a bit of background on penicillin G. This is, of course, the antibiotic that was discovered by Fleming and soon became one of the most important antibiotics in that golden age of antibiotic discovery. And because it was used very early, this is one of those antibiotics that has probably had the largest chance to contaminate the environment, if you will, and possibly select for interesting bacterial interactions. And it's nice that this is, again, a natural product antibiotic, unlike the sulfamides, because this means that we know that it is actually made out of discrete molecular units that could fit into essential metabolism very well. And what I mean by that is that it's synthesized out of a phenylacetic acid group, cysteine amino acid, and a valine amino acid. So you can very easily imagine how this could be recycled in essential metabolism by bacteria. And finally, we know quite a bit about how it's resisted, most commonly through these enzymes called beta-lactamases, which break this beta-lactam ring, or which prevents this from being antibacterial anymore. And so what I want to do is come up with a hypothesis for how antibiotic metabolism could happen. What I came up with is this retrobiosynthetic hypothesis, where probably following a sort of safety first philosophy, bacteria would inactivate antibiotics through a resistance enzyme, that they would probably use catabolic enzymes to break down these natural products back into the molecules that they're made of that can easily fit into essential metabolism. So in the case of penicillin, that's again these amino acids and phenylacetic acid. So I want to introduce the players of this I'm going to be talking about. So these are the 4 soil isolates that I focused on. And I'm going to be taking a bit of a shortcut and referring to them by their code names of ABC for antibiotic catabolizer 02, 07, 08, and 10, because I don't focus much on the significance of these different taxa. But I do want to point out are 2 things. First off, that these are all taxa that have close relatives that are human pathogens. So anything we learn about antibiotic catabolism and how resistance fits into that could have direct relevance to clinical isolates. And number 2, these are really cool bacteria, because you can see that in these growth curves, the color indicates how much carbon source was added, where gray is nothing and blue is 1 gram per liter. And in this case, these were grown in minimal media where the only carbon source is penicillin. And so you have this really great but backwards relationship where the more penicillin you add to these cultures, the better they grow. So the way that I set about with my collaborators, figuring out exactly how these bacteria metabolize penicillin, started with genome sequencing and RNA transcriptomic sequencing. And what we did was we grew these strains in minimal media with penicillin as a sole carbon source, and we compared the transcriptome of those strains against them when they were grown with something like glucose or histidine as a sole carbon source. So these are carbon sources that fit directly into central metabolism. And what happened when we looked at the significantly upregulated genes under these conditions was that all 4 strains kind of showed this very interesting pattern where all 4 of them had highly upregulated beta-lactamases in blue, which makes sense. That's sort of the safety first approach. They would be dead if they didn't have these beta-lactamases. And then also really interesting, they had a upregulated phenylacetic acid catabolon in each case, shown in green. And now, of course, phenylacetic acid is actually one of the components that is used in the biosynthesis of penicillin. And so that really does suggest that after inactivating the antibiotic, somehow these bacteria are getting at that phenylacetic acid, and that is maybe what the underlying carbon source is. And that's what made these other upregulated genes particularly interesting in the red, which had very vague but interesting annotations like hydrolase or amidase or butyrhydrolase, as well as a few permeases. And so we think that that is where kind of the heavy lifting of penicillin catabolism is happening. And so I decided to focus on the strain ABC07 in more detail to see exactly what might be going on. So we repeated these transcriptomics experiments and again saw that all of these target genes were upregulated during growth of penicillin. But interestingly, when we grew the strains with benzyl penicillic acid compared to glucose, we saw that this first beta-lactamase gene was no longer upregulated, which makes sense because the beta-lactamase target is this beta-lactam ring, which is already broken benzyl penicillic acid, so it's no longer an antibiotic, and you no longer need this beta lactamase. What was really interesting and exciting was that when we looked at upregulated genes when phenylacetic acid is the carbon source, again, the phenylacetic acid catabolon was upregulated, but this entire potential operon, which we started calling the penicillin utilization operon or put operon, was completely downregulated. And so this really suggests, again, that this operon might be what's responsible for freeing that phenylacetic acid to be used as a carbon source. And just to confirm that this is important, we made a couple of knockouts. We first made a knockout strain where we disrupted this whole put operon. We made a second strain where an essential step of phenylacetic acid catabolism was interrupted. Both of these strains grew perfectly well with glucose as a carbon source, but very notably, neither of them could grow using penicillin or benzyl penicillic acid as a carbon source, really indicating that these two functions are essential to catabolism. And so, to kind of put a long story short, I found that this PUT1 enzyme, which had an annotation of amidase, appears to be particularly important in this function. What I'm showing you is a series of LC-MS experiments that I performed on reactions where the purified PUT1 enzyme is incubated with benzyl penicillic acid. We can see is that in this positive control, this is an interesting enzyme called penicillin amidase that can actually use this as a substrate poorly, because you see that the benzyl penicillic acid is partially degraded over time. But in the PUT1 amidase reactions, I saw that at this final time point, it almost completely degraded benzyl penicillic acid. We have other experiments suggesting that, again, it's attacking this amide bond and releasing phenylacetic acid. So with these experiments, we came up with this model for penicillin catabolism, ABCO7, and we think it might hold for the other ABCO2, ABCO8, and ABC10 strains I showed you, where penicillin can enter the periplasm through a porin protein, where it interacts with secreted beta lactamase enzymes, which inactivate penicillin to benzyl penicillic acid. And we think maybe this gene, which is annotated as an efflux pump or a permease, could maybe have a role in allowing that benzyl penicillic acid to go from the periplasm into the cytoplasm, where we think these amidases like PUT1 and PUT2 localize, and those are probably ultimately responsible for removing phenylacetic acid from this compound, allowing it to go to central metabolism and act as a carbon source. And so this is a really cool thing, and what we want to do now is figure out, we have this pathway that can explain very well. Can we make this useful? So we thought, could we actually turn E. coli into a penicillin catabolizing bacteria? So I found a strain of E. coli which has a phenylacetic acid catabolon, which is one of the necessary components of this pathway. And what I did was I gave it a beta lactamase expressing plasmid, then had it express this penicillin amidase enzyme, or gene penicillin amidase. And it's actually, again, this positive control I used earlier. And this is an enzyme that's used industrially to remove phenylacetic acid from penicillin so that we can make semi-synthetic penicillin analogs like ampicillin or amoxicillin. And so it has that necessary function of removing phenylacetic acid from penicillin-like molecules. And what I found really excitingly was that, indeed, this E. coli strain could now grow in minimal media with penicillin as a sole carbon source. And once again, I saw this really neat dose-response relationship where the more penicillin I added to minimal media, the better the growth of that E. coli strain was. And so let's kind of wrap up this, kind of interesting look at bacterial antibiotic interactions. I want one of your takeaways to be that there are these really cool bacteria out there that can actually use antibiotics as a sole carbon source. And that I think that this probably occurs more generally through a retrobiosynthetic set of reactions where catabolism happens by essentially reversing the biosynthesis and turning an antibiotic into smaller units that can fit into central metabolism. What was really interesting was that in ABCO7 at least, resistance and catabolism were intrinsically linked. They are physically linked because there is a beta lactamase gene synthetic to these important amidases, They're transcriptionally linked because these all seem to operate as an operon or transcribe together, and it's metabolically linked. So one thing I didn't show was that PUT1 does not appear to use penicillin as a substrate, but it does use benzyl penicillic acid, which means that metabolically it needs a beta-lactamase to act upstream of it in order to function. So in this case, catabolism and resistance are inextricably linked together. And then finally, one thing that we can learn by studying this is how to make potentially useful bacterial strains, so things like E. coli that can actually potentially bioremediate penicillin from the environment, or maybe be used to discover new and better penicillin amidase genes from the environment. And so with those two stories done, I want to finish up with one quick story that is about this really useful method that can be used to study antimicrobial bacterial interactions, and how I made some changes to that method to make it much more efficient. And so one of the common ways of studying microbiome and antimicrobial interactions is through high throughput sequencing, where you take a microbiome or a source of metagenomic DNA, extract that DNA and sequence it. And this has been a very powerful technique for studying antimicrobial microbiome interactions, but it has a weakness, and that is when you get a sequence result out, you have to take that and compare it to a database of annotated genes in order to get at what functions are present in that environment you just sequenced. And this is very useful for very well-studied genes and enzymes. But when you find a novel gene that has a low identity to anything that's known, the annotations that you can get out are much poorer in quality. And so this is kind of shown in that antibiotic metabolism story, where we had these really interesting potential penicillin amidases, but they had very vague annotations of hydrolase or amidase when we started that experiment. And so one way to get around this problem with sequencing is to use these really useful tools called functional metagenomic libraries. And so what these libraries are these physical libraries that you can make by taking the metagenome, shearing that DNA into gene-sized pieces and ligating that in mass into expression vectors, and then transforming those into E. coli. So that gets you this sort of physical library of millions of E. coli cells, where each E. coli cell is hopefully expressing a gene or multiple genes that originated from your microbiome source. And what this allows you to do is it allows you to basically free these genes from their native context, force their expression, and study them. So you can study them in this very intractable E. coli organism. So for a good example of this is you can take this library and flavor it, for example, on an antibiotic like penicillin. And any colonies that grow up in that condition must have originated from an E. coli cell that managed to capture something like a beta-lactamase from the environment. And at that point, it's trivial to extract and sequence the captured pieces of DNA. And now you can get something out of your selection that has a very, again, maybe not useful annotation of just a hydrolase. But because you know it came from this penicillin selection, you can now go through and say, well, I think this hydrolase is actually a beta-lactamase. And that's a way you can find these really novel genes and enzymes based on their function and not needing to rely on existing annotations. So again, this is a very useful method because it's a high throughput method. You can select or screen on gigabases of DNA at a time. And kind of the most important part of it is that it's sequence naive. You don't need to know what you're looking for when you do a selection. You just need to know what function you want to study. And you don't need to know the sequence of the enzymes that you're trying to find coming out the end. And also very usefully, because you're doing this in E. coli, it's now a cultivation independent technique, where another big bottleneck in microbiome studies is the fact that something like only 1 to 10% of microbiome derived bacteria are easy to culture in the lab. And so this is a really useful method for getting around these bottlenecks. But it also has its own problems, and that is the actual preparation of these libraries is very inefficient at times. And so this inefficiency basically prevents researchers from making these libraries out of rare samples or samples that have low biomass. And this inefficiency comes from two steps. In many cases, the shearing of DNA to get gene-sized pieces is performed using things like covariance sonicators. And these are very DNA greedy. You need to have very high input masses of DNA to function. And so this is kind of what directly limits you to using plentiful microbiome samples, or else pooling small microbiomes together to get a high enough mass. But in the process, losing any sort of information you have about individual small microbiomes. Then another step that limits this method is the cloning of these randomly sheared fragments into an expression vector. Because you don't know anything about this DNA, you have to use something like blunt ligation. And we know that blunt ligation is very inefficient. You need to use a large excess of inserts to vector. And even if you do that, most of the inserts you have are wasted and don't get successfully added into a plasmid. So in short, you need to have a lot of DNA that goes into this method, and most of it is wasted in the cloning. And so what I set out to do was figure out a way to make this a little bit better, essentially by copying off a lot of innovations that have occurred in the area of high throughput sequencing and cloning advances that have been made in the last few years. And what I came up with is this thing that I'm calling mosaic ends tagmentation assembly, or meta assembly. And so the first kind of innovation is in the shearing of the DNA. And so this copies very directly from high throughput sequencing, where folks have discovered that you can use transposase enzymes to break your DNA down into small pieces instead of something like a covariance sonicator instrument. And the advantage of this is that you can go from having a 5 microgram input requirement down to something like just 50 nanograms or 5 nanograms, so orders of magnitude less DNA are required. And so you could basically have this tagmentation fit directly in with the existing library preparation protocol, where you blunt the ends and blunt ligate into a vector. But if you're doing this tagmentation on a 50 nanogram sample, then that's going to be essentially completely used up, and it's going to be wasted with blunt ligation, because this is such an inefficient process that even if you can successfully fragment a low input mass sample, you're going to get just a tiny library as a result of it. And that's really kind of the second part of this method comes in, the assembly. And there's this really kind of, I think, cute thing that we figured out where, it's known that you shear DNA by tagmentation, the transposase enzyme installs known oligos on either end of the fragments of DNA. And it turns out that these oligos in their shortest version are just 19 base pairs long. And that turns out those sequences are perfect for molecular assembly techniques. So things like Gibson assembly or hi-fi assembly, because we know that every insert produced by tagmentation is going to have these sequences on the end. We can essentially prepare the expression vector to have those same matching sequences, and then use these assembly cloning techniques that rely on hybridization between insert and vector to much more efficiently, in theory, take these inserts and clone them into a vector. And this is important because, again, tagmentation allows you to use low input mass, things like 50 nanograms of input DNA, but blunt ligation would just waste that. And so the idea here is that we can pair assembly with that tagmentation. We can actually make much more efficient use of that 50 nanograms that we put in the first place. And so to sort of illustrate this boost in efficiency, and I show you a couple of examples of making libraries using the meta-assembly method. And so this first one just started with soil that I extracted from metagenomic DNA, and I used 5 micrograms of metagenomic DNA to make a library. So this is, again, kind of that standard minimal input used to make functional metagenomic libraries in the sort of classical protocol. And the library that I got out of this with some data in the middle turned out to be 712 gigabases large, which I'll tell you is very good. And to put that into context, when showing you are the library inputs and library sizes from 6 other published works using that classic method, where the mass of DNA used to make these libraries is shown in squares, and the resulting library that was prepared is shown in circles. So in many cases, these other manuscripts use between tens of micrograms to hundreds of micrograms of DNA to prepare libraries that were tens of gigabases to hundreds of gigabases large. And to put the meta-assembly library in context, I used just 5 micrograms of DNA, which is a fraction of the rest of these manuscripts, to prepare a 700 gigabase large library, which is essentially the same size of all 6 of these prior studies combined. So that's what I mean when I say there's a tremendous gain in efficiency. And one of the really cool things that we did with this library was use it to select for resistance genes to streptothricin. So again, I want to find, without knowing the gene I'm looking for, what genes in this library can give resistance to streptothricin in E. coli. And I found a lot of really interesting things. Genes like aminoglycoside acetyltransferases, or these genes that we decided after looking at them belong to the streptothricin acetyltransferase family of enzymes. What's kind of neat was that a lot of these genes that confer streptothricin resistance are actually synthetic to other antibiotic resistance genes. So in this case, you have an acetyl transferase that's synthetic to a gene that might give resistance to fluoroquinolones, or a pair of aminoglycoside transferase genes next to each other. And kind of what highlights the utility of functional metagenomics is when I looked at one of these genes that we call a streptothricin acetyltransferase. And so what I show you here is a enzyme phylogenetic tree that we've populated first with the known streptothricin acetyltransferases shown in red, as well as in pink, if you can find them, as well as populated with the experimentally derived streptothricin acetyltransferases from our selection, and the other apparently any other enzymes that appear to be homologs. What I want to emphasize is that these red circles are really represent the known extent of streptothricin acetyltransferase enzymes. What's really cool is that the ones we discovered with this library appear to represent new branches in some cases. And to emphasize again that if we had just sequenced this soil sample without doing the functional metagenomic library step, we could have found this gene, and it would just be annotated as a gene acetyltransferase because it has very low percent identity to any other well-annotated enzyme like this streptothricin acetyltransferase enzyme. But because we coupled this with functional metagenomics, we know that this enzyme is not just a random acetyltransferase, it is actually a streptothricin acetyltransferase. So that really kind of illustrates the utility of functional metagenomic libraries. And so what I did next is I want to show another library that really gets to this idea that the added efficiency you get by preparing libraries with meta-assembly allows you to use very low input mass microbiomes. And so it turns out that Northwestern campus where I work has a lot of Canada geese. And so I got permission to just scrape up some of their goose poop off the sidewalk essentially, and use that as an input source for a new library. And so for this, I used 300 nanograms of metagenomic DNA. So again, this is an order of magnitude lower than the bare minimum that you can use in the more traditional library preparation method. And this resulted in the preparation of an almost 30 gigabase library in E. coli. And what I'm showing you here is those same 6 manuscripts where I basically measured their they were able to prepare using 1 microgram of DNA. And it turns out, for the most part, all these groups get on average 1 gigabase of library per microgram of DNA that you use. And in contrast, across the 4 libraries that I prepared for this manuscript, as well as others I've since prepared, I get almost 100 gigabases of library per microgram of DNA, probably closer to 80 gigabases per microgram. But again, that's something like an 80-fold increase in efficiency, which is why these kind of low biomass inputs or these low DNA input experiments are possible. And so to again kind of show why this sort of research is cool, why this is a useful tool in general, I selected the library that I prepared from the goose microbiome on a couple of antibiotics, including tetracycline, which selected mostly for efflux pumps, but really interestingly, essentially all the efflux pumps we found were synthetic to mobile genetic elements, so things like transposons, phage genes, potential plasmid initiators of replication, really showing that in the goose gut microbiome, at least, a lot of tetracycline efflux pumps are ready to be spread among different bacteria. And I also selected for colistin resistance in this library. We found a lot of genes that are involved in manipulating the outer membrane of bacteria, which is the target of colistin, including this MCR-5.2 homolog, which is a homolog of that recently discovered, somewhat frightening MCR1 class of resistance enzymes. That basically shows again that the goose gut microbiome encodes resistance enzymes that, if they spread into pathogens, could make them resistant to one of these last lines of antibiotic treatment we have available. And so to kind of wrap up meta-assembly story, I want you to remember that functional metagenomic libraries are this really useful tool for sequence-naive and cultivation-independent discovery of new functions for microbiomes. And this works especially well for antibiotic resistance studies. And that if you combine fragmentation, DNA sharing with molecular assembly protocols, you can get something close to 100-fold boost in efficiency and basically be able to prepare these libraries from microbiomes that are 10 to 100-fold lower in biomass than previously available. And so with that, I just want to wrap this up and remind you of the 3 stories I told. The first being this story about chloramphenicol reductases and how we can look at housekeeping enzymes as being an early step on the evolution to antibiotic resistance, and how there are these really neat bacteria out there in the environment that might be responsible for recycling antibiotics that are produced in the soil, and how we can use the pathways of those bacteria using antibiotic catabolism to inspire potential bioremediation, bacterial strains, or industrially useful enzymes. And finally, like I just talked about, there's this really great tool of functional metagenomic libraries that I've made along with collaborators about 100-fold more efficient. And this is a really great way to study these potentially novel antimicrobial bacteria interactions. So that I want to thank my collaborators. A lot of help when I was in the Dantas lab with these 4 grad students helping on the antibiotic catabolism story, and these 4 really talented undergrads who helped on the first chloramphenicol reductase story. I also want to thank my collaborators at Argonne National Labs for working on the crystal structure and enzyme purification for the second half of the chloramphenicol story. I want to thank the Kelleher group, who have had a lot of useful conversations when I work with them. And then also my collaborator, Erica Hartman, and her grad student, Alex McFarland, who did a lot of help on that meta-assembly story with the annotation and computational analysis of the functional metagenomic libraries. And with that, I'd be happy to take any kind of questions.
Right. Very cool.
Thank you.
Yeah, the last part was awesome. I look forward to that paper when it's out.
Hopefully soon.
Yeah. So for everyone who has a question, please type it in the live chat box on YouTube, and I will take them for you. Meanwhile, I have a couple of questions for you, Terence. So one is about the first part that you talked about, the housekeeping genes evolving into resistance genes. I was wondering if you just take multiple isolates from nature, will you have a difference in expression or enzymatic activity of these housekeeping genes among, considering that one strain is resistant but the other one is susceptible? Will you see a difference if you just take some strains from nature?
I think, yeah, that's a really good question, and a way to look at how the kind of native context of the housekeeping gene matters for whether or not it can give resistance. And so when I chose these NFSVs to study, I didn't actually think that the H influenza one was going to be the real winner, because the paper that came from says that H influenza strains can reduce chloramphenicol, but it does not actually give those strains resistance. So in the native context, even though we know that that enzyme has the ability to give resistance, those bacteria haven't essentially, I guess, discovered that upregulation would give resistance. And so it's only by taking that gene out of its native context and overexpressing it in E. coli do we see this antibiotic resistance. So, that's one of the barriers between having the housekeeping gene become a resistance gene, is the level of expression, probably.
So there is some level of contribution to the resistance in your experiments because of E. coli itself?
That's a good question. So I don't know if, I stuck with using E. coli as a host because it's the most tractable thing out there.
Right.
Whether or not there's something unique to E. coli that means that it can get resistance by this mechanism that other bacteria can't, I would guess it isn't. One of the things about Smallpox influenza is that it is almost, that taxa is almost uniquely susceptible to chloramphenicol. In many cases, chloramphenicol is a bacteriostatic antibiotic, so just stops growth, but in Haemophilus influenza, closely related species is actually bactericidal, so it kills those bacteria outright. So it might be that they do have some particular physiological quirk that makes them extra susceptible. And so maybe that's why the transferase E. coli is needed to give resistance.
Okay, that's interesting, Mary. Okay, and so I have another question about the last part where you see that a lot of transposons are getting picked up in the selection where you're selecting for the resistance. So do you think it's because of the transposons evolving into efflux pumps which are driving the resistance, or is it just their inherent feature of those transposon genes?
Right. So about seeing this these mobile genetic elements being right next to these resistance genes. So I don't think that it's anything that's particularly to the efflux pumps.
Okay.
This kind of highlights one of the cool things that you get out of functional metagenomic libraries, is that when you have a selection, oftentimes the insert has room for multiple genes, so 2 or 3 genes can fit on that randomly grabbed insert. And so it gives you kind of a snapshot of the neighborhood of the resistance gene, which is how we could see that there are these mobile genetic elements. I don't think that there's anything particularly special about the efflux pumps that means that they're next to these elements. Mostly this just reflects probably the fact that inside the goose microbiome, there is a selective pressure for tetracycline resistance. So, bacteria that do have resistance oftentimes might have it because they picked it up from other bacteria based on the presence of these mobilizable elements.
Okay. Yeah, it's just the presence of them right next to these genes is quite interesting.
No, I think it's really neat. Reflecting again, probably antibiotic use has encouraged environmental bacteria to basically put together these resistance cassettes that have, a tetracycline resistance gene next to a regulator, sandwiched between mobilizable elements.
Cool. Okay, so if you have any other question, people, please put it in the live chat box. If you are listening to this talk at a later time, and if you have questions, you can either put it in the comment section or reach out to Terence directly. Okay, I don't see any questions popping up right now, so I think we will call it off. Thank you, Terence, for a wonderful talk.
Thanks for having me.